Hi everyone, we're back with lecture 28, video number 3. So let's talk more about photosynthesis. We are into the light reactions, step 3 on this slide. We left off talking about reduction potentials, and I said I'd tell you how reduction potentials get high enough to reduce or to oxidize water, to pull an electron from water. So let's look at reduction potentials in this slide. So on the y-axis, you see reduction potentials, and this is the same scale we've seen before. Now, here's P680. When it excites an electron up to P680 star, which is the excited electron that comes from P680. So P680 excites an electron by absorbing light. That excites it up to a primary electron acceptor. That has a reduction potential of minus 0.8. So it's really low. Okay? So in other words, that electron wants to go somewhere. It can be pulled by anything that has a reduction potential higher than minus 0.8. Minus 0.8 is as high as we've seen so far in class. Okay? So that's key. Now, the next thing, after that electron is excited by light and removed from P680, the reduction of potential of P680 after donating the electron is around 1. That is really high. Remember, the biggest electron potential we've seen so far is a reduction potential of oxygen, which is about 0.8. Well, look at the scale here. Water and oxygen are two sides of that reduction reaction. If we were looking at oxygen being reduced to water, we'd be looking at a reduction potential of 0.8. So to pull an electron away from water, we need a reduction potential higher than 0.8. If it was lower than 0.8, nothing would happen. We wouldn't pull an electron from water because there's a greater tendency for oxygen to pick up an electron. But if you have an electron, or a reduction potential rather, greater than 0.8, which we do here, we've got 0.1, we can pull an electron from water. So we use a manganese center. Manganese is a key metal ion that's in the reaction machinery in photosynthesis. And that electron will flow through that magnesium center, and it will flow to P680 from water. So, we excite an electron, we create a gap in P680, and that gap, or that hole that's created, we call it a hole, that has a reduction potential of 0.1, and that allows us to pull an electron from water. Pretty cool, huh? So we end up making oxygen, and the protons that came from water end up getting pumped and becoming part of the proton gradient. Now, when the electron in P680 is excited to a higher energy state, P680 is more easily reduced. This is really the take-home of that starting process in photosynthesis, right? Excite an electron, create a hole. P680 then can be reduced, or in other words, can pick up an electron. In this case, can pull it from water. Very cool stuff. So here's the image from the book. That hole created in 680 oxidizes water. All right, so it's a 2-electron oxidation ultimately. Pull an electron one at a time, but we pull 2 electrons. We actually pull 4 in total to make a full O2 molecule. 2 electrons will give us a half of an O2 molecule, and obviously we'll pull another 2 electrons and another oxygen from water. So it takes 2 waters, and we'll make a full O2 molecule. Now we're on really to step 4, electron transport chain. So the primary electron acceptor, once it gets that electron, it has a potential of minus 0.8, so we said it's going to easily flow to other places with bigger reduction potentials. And that's exactly what it does. It flows down the electron transport chain through electron centers that are shown here similar to the electron transport or the electron centers that we saw in the electron transport chain in oxidative phosphorylation. This schematic kind of shortcuts the process because in addition we are pumping protons when these electrons flow, and it's pumping just by the same mechanism that we saw in oxidative phosphorylation. And then that electron gradient is used in step 5 to make ATP. So we don't show all those steps here in detail. Here's the picture of what it looks like in the thylakoid. This is a nice schematic. So remember, thylakoid space is the space into which we pump protons. So here we're pumping protons inside the thylakoid. Outside is the stroma, which has low hydrogen concentration, proton concentration. So by diffusion, simple diffusion, that proton will flow, the protons will flow back through ATP synthase to relieve that chemiosmotic gradient, the difference between high protons in the middle, so a lot of plus charges in the middle of the thylakoid, in the thylakoid space, and they'll flow through ATP synthase and make ATP just like we saw in oxidative phosphorylation. So like I said, there are a lot of parallels. The thing we add, light, that it starts the process. Rather than pulling electrons from glucose, light excites electrons that are pulled from water. The other part of the process that we talked about is photosystem 1, shown here, embedded in the membrane. That excites electrons again so that they can flow down another electron transport chain that does not pump protons, but dumps those electrons on NADPH. Then the ATP and the NADPH can be used in the dark reactions in the Calvin cycle. And that is where we take CO2 and we fix it into sugar or into carbohydrates. So this is step 5. I already talked about it, and I said that we just don't see a lot of details here, but step 5 is ATP being created by the chemiosmotic gradient that came from pumping protons. Finally, here is a nice comparison. So if we had the mitochondrion, which is shown on the left side of this slide, and we had the thylakoid shown on the right side of the slide. In both cases, we have a membrane that contains an electron transport chain that pumps protons. In both cases, we have ATP synthase through which the protons flow to create ATP, right? So those are the same in photosynthesis and in the mitochondrion. Really cool. The differences are, in the mitochondrion, we have an intermembrane space into which we pump the protons through the inner membrane, and we pump out of the matrix. On the other hand, in the chloroplast structure, we pump from the stroma into the thylakoid space. So make sure that that's clear. It's a little confusing just because we have the thylakoid space and the stroma on the right side. So it kind of looks like we're pumping from back through ATP synthase from the thylakoid space to the stroma. But before we pump through ATP synthase, we pump from the stroma to the thylakoid space, and then the protons flow back from the thylakoid space to the stroma through ATP synthase and make ATP. Okay, so just be clear on that. The chloroplast structure is shown on the right, the mitochondrion structure shown on the left. That's it for this video. Thanks for watching. We'll see you in video number 4 of lecture 28.